Right, we might get started. Welcome everyone to this afternoon's symposium session on the topic of Are plant traits a useful indicator of restoration targets? My name's Mimi and I will be your chair for this afternoon. This is actually a sister symposium of a session that was held 2 weeks ago at the European SERA conference in Iceland, and I'd like to thank the invited speakers for attending, as well as welcome the high school students that are participating in the QUT Vice Chancellor's STEM program that have been measuring their own plant traits this week. So to begin with, our first speaker is Angela Moles, who will be providing an introduction into the theoretical understanding of global pattern plant traits. Angela leads the Big Ecology Lab in the Evolution and Ecology Research Centre at the University of New South Wales in Sydney. Here she is.
Thanks, Mimi. I somehow feel disappointed not to be in Iceland now, though. All right, so I'm going to change a little from the global aspect, but we are still talking about the big-scale patterns of plant traits. And the thing with plant traits is that they're often quite easy to measure and they can tell you important things about how species grow and reproduce. And so people have seen them as a sort of a holy grail. That was kind of the basis of this symposium, that we could use to give insights into the effectiveness of restoration efforts and to help inform species selection for planting efforts. Now, the trouble here is that the traits literature is absolutely immense and it grows so fast you wouldn't believe it. So instead of having a holy grail, we can often end up with something more like the goblet of fire that takes you like dark places where you didn't want to go. So what I'm going to do with my seminar today is really get on a soapbox about two things that we now understand differently to what we did in the past and which bug me because people still think the old way. So I want to just do that. So the trait I'm going to focus on most is seed size. Really is my favourite trait. Globally, there are species spanning 11.5 orders of magnitude of variation in seed size. So the smallest seeds on Earth weigh less than 1/1,000,000th of a gram. And some of those are orchid seeds, like the vanilla seeds that you get, you know, those little black dots in the ice cream. So they weigh less than 1/1,000,000th of a gram. There's not even enough energy in them to get a seedling going. They need help from their symbiotic fungi. At the other end of the spectrum, we have the double coconut, which I love because it looks like a giant bottom. It even has a little furry patch in the middle. And they can weigh up to 27 kilograms. And even if you look within a particular ecosystem, you still see that the species making the smallest seeds is usually making seeds 1,000,000 times lighter than the seeds made by the biggest seeded species that's growing right next to it. So there's this massive variation in seed size. And I'd say around about 2000, we really thought we understood seed size. And the main thing that we thought was going on was that it was a trade-off between making a lot of small seeds that had low survival abilities versus making a few big seeds which made much better seedlings that were much more likely to survive. And we had a bunch of evidence for these things. But what went wrong here is that we got a little fuzzy in our thinking. We thought, well, you know, the lifetime fitness, those would counterbalance out. But if you actually want to know the lifetime fitness, you need to do more than just think about seed production and seedling survival. You need to think about how many seeds the plant gets to produce during its whole life and how many of those seedlings survive all the way to adulthood. So this was our first piece of evidence, the seed size number trade-off. It's quite remarkable. Across all these species, we have a slope almost not significantly different to -1. So it's a direct trade-off between making bigger seeds and making more seeds. But the units there that I've got circled are per square metre of canopy area per year. So that's saying if you've got this much canopy, you can either make, you know, a lot of small seeds or a few big seeds. But we want to know the lifetime seed production. Now, that wouldn't affect this if there was no relationship between seed size and canopy area or plant lifespan, but there is. Big seeds tend to come on big plants and small seeds tend to come on small plants. So if we, instead of looking at the number of seeds per square metre of canopy area per year, if we look at the number of seeds produced per adult plant per year, the slope drops down to -1/3. So that's a lot less steep, and it's become a lot more noisy, right? But this is still on a per year basis. And it turns out that bigger seeds come on bigger plants that have a longer reproductive lifespan. So these plants might be producing seeds over like 100 years. So they get a lot more chances than some of the little herbs at the other end of the spectrum there. And once you look at the relationship between seed size and lifetime seed production, there isn't one. So small-seeded species do not have higher seed production, and that's something that you see misunderstood in the literature all the time. So that's kind of interesting, because we've looked at this top part of this and we found that that's wrong. So that's kind of cool, right? In science, when you assess something that we think we've always known and you go, oh, it doesn't work like that, then you get kind of interested. So if we have no relationship between seed size and lifetime seed production, but we have higher seedling survival to adulthood, then we'd get this runaway selection for double coconuts, which I find kind of conceptually amusing, but it's clearly not happening in the real world. We do have all of those different sizes of seeds out there coexisting. So let's have a look at the other side. This is the relationship between seed size and seedling survival through their first week. And you can see those big seeded species, they do have higher rates of seedling survival. But that's through their first week of establishment. And, what we need to know is seedling survival to adulthood. And it turns out that the seeds that we have, their advantages of those big seeded species are transient. They've got a certain amount of extra resources they can use for that seedling to grow, but they wear off. If it's a really big seed, it might take a year or two for that advantage to wear off, but it does wear off. So, if we look at the relationship between the number of surviving individuals and time, and we're looking at a small-seeded species, they sort of die at a fairly constant rate through time. Now, if we put our big-seeded species on there, that will initially have a lower rate of mortality, so there'll be more individuals surviving, and then after a while they start dying at the same rate as the small-seeded species, and there's lots of experimental evidence for that. Now, if they reached adulthood at the same time, then the large-seeded species would have more surviving offspring. But large-seeded species with those large plants and those long lifetimes also tend to take a long time to become adults. And so what's happening is that the small-seeded species reach reproductive maturity. Oh, sorry, I wanted to get a pointer. No. The small-seeded species reach maturity earlier than the large-seeded species, and the evidence is kind of scarce on this, but it looks like there's no significant difference in the proportion of seedlings surviving to adulthood. So, we had this, it was all so clear, but that was wrong, and that was wrong, so we've actually got, a whole bunch of different things going on. And if you'd like to read that through more slowly, it is in the Harper Review that I published at the start of this year. So what happened here really is we were looking at this trade-off here. But you know that thing about the elephant and the people with blindfolds on all thinking they've got different bits? We looked at just a little part of the picture. What's really going on is this. So to simplify that, plant traits are best understood as part of a species life history strategy. So at one end of the spectrum, we've got our nice big oak tree here, making, quite big seeds, but it's got a great big canopy, and it's going to make those seeds over maybe a couple hundred years. Down here, we've got a bunch of herbaceous species, have much smaller canopies, smaller seeds, and they're only probably going to be annual or biennial in most cases. So we've got a spectrum here from fast life histories to slow life histories. Okay, now the next thing I wanted to tackle is something that people often think might go along with this spectrum, is a seed dispersal distance. So who thinks small seeds would go further? Who thinks large seeds would go further? Okay, so basically in the literature there's loads of stuff saying, well, small seeds ought to disperse further, But one of my PhD students, Fiona Thompson, compiled a whole bunch of data from the published literature on seed dispersal distances achieved under natural conditions. And what she found was that both mean and maximum seed dispersal distance are positively related to seed size, which is quite surprising, right? These big, heavy seeds are managing to disperse further. And that even holds within the wind-dispersed species. In fact, it's more tight within the wind-dispersed and unassisted dispersal species than within the species that are carried around by animals. So that's kind of interesting, because it turns out that seed mass is a good predictor of how fast seeds fall to the ground. But in the real world, dispersal distance depends on a whole lot of other things, like the texture of the canopy and how high up they're being dropped from and all sorts of things like that. And in fact, if you look at the relationship between plant height and seed dispersal distance, you find it's much, much stronger. There is no relationship between seed size and seed dispersal distance. So if you want to know how far a seed is going to go, look how tall the tree is, not how big its seeds are. So again, we were looking at a part of the picture, but not the whole thing. And just in case you're thinking that it's my science that's a bit funny, it was quite nice. Last year, Carol Augsburger and other people from Barrow, Colorado Island published basically the same thing based on 12 neotropical species that had wind-dispersed seeds. They didn't cite us, but so there you go. It's a completely independent discovery of exactly the same thing. All right, so implications for restoration and also for understanding weed invasions. Again, the traits of species have been seen as some sort of a holy grail, like the traits of the ideal weed back since, Baker has been something that people have been trying to figure out. And it's been very murky, right? Some studies will show that this is better and some will show the other is better. It's all over the place. And I just want to sort of share a little insight into why I think that has been. Generally, while you're out in the field, it's very human to go, oh, this thing is such a successful weed, it must be because it has bigger seeds than anything else in my site. But if you look at what's happening in all these different sites, remember how we've got that 6 orders of magnitude of variation within a given site. There is no best seed mass. If big seeds were better, we'd have those double coconuts all over the planet. So remember that there is no fundamental best seed mass. So if you find yourself looking for the trait of what would be best to plant, there is no easy answer under this stuff, because if being small was better, everything would be small. So that applies to basically all of the traits. What we can do, however, is have a think about how trait distributions change with environmental conditions. So, for instance, if we have an environmental gradient like rainfall, or here are some real data, these are seed sizes changing with net primary productivity, so you get a shift in the distribution as you go across those gradients. And this is quite close to some of the stuff that Daniel Laughlin said this morning, that basically we can use those existing data, use some clever multivariate statistics to generate a model that gives us a predicted distribution of a trait under given environmental conditions. Then what we can do is run that model once for the original conditions and then run it again for the present conditions. Because often we're looking at sites where there's something different, be that climate change or increased disturbance regimes or decreased burning frequencies, increased nutrients, that sort of thing. If we overlay those two distributions, that gives us 3 trait zones. The red space is trait space that used to be viable under the old conditions, but isn't viable anymore. So any native species that had traits in that part of the spectrum is going to be at risk of local extinction. And it also means that unless you can change the climate back, or the nutrient, or the disturbance regime, you're going to really struggle to get those things to survive in a site to which they're no longer suited. So that kind of switches the emphasis away from the traits and back to the environmental conditions that they go with. The orange zone in the middle are the native species that can still fit the current conditions, and the degree of overlap there just depends on how big of a change you've had in your ecosystem. The third zone there, the yellow zone, is kind of interesting space. That's trait space that has recently become viable. Wasn't there in the old times, and there may be no native species that have traits that are suited to that, or not at least in the local community. So this is something where you might get introduced species coming in, like around the Sydney region where I'm from. We get loads of introduced species where we've added more nutrients than our native plants can deal with. And people can pull out those introduced species, but they're not actually fixing the fact that the native plants just don't like growing with that many nutrients. So this is an opportunity, I guess, for introduced species to come in, but it's also, if you want to start thinking about novel ecosystems retaining their function under the new conditions, you might want to start thinking about things like species translocations, but that all gets horribly controversial. Okay, so 2 big take-home messages. First is we need to watch out for elephants. It happens all the time that we're just considering one little part of the picture. So if you want to understand the fitness advantages of a plant trait, you need to do it over the whole lifetime, not just with respect to one little piece of the puzzle, or you can come out with the wrong idea. Second conclusion is it won't be as simple as bigger is better, but traits combined with environmental data can be incredibly important for understanding restoration efforts.
No time for questions. So next we have Jennifer Firn, who is an Associate Professor at the Queensland University of Technology and also Editor-in-Chief of the Journal of Ecology and Evolution.
Thanks, Mimi, for the introduction. I'll just... Oops. Oh, there I am. Exciting. There we go. So that should work. I did it as a PDF, so I just want to check if it's working. So thank you to all the speakers, and thank you, Mimi, for the introduction. So we're going to move from seed traits now to leaf traits. So we're moving up slowly, probably to multiple traits with Dan at the end. So kind of has a purpose or some kind of goal that just worked out that way. So I'm going to talk today about leaf trait responses in an experiment where we looked at leaf trait response to fertilization and also changes in herbivory. So why leaf traits? So there has been a very strong focus on leaf traits, particularly in herbaceous communities such as grasslands. And there's really good reasons for focusing on leaf traits as a potential measure of the function of communities. And that's because plants invest in photosynthate and mineral nutrients in the constructions of their leaves. And leaves, in turn, assist plants to capture light to produce more photosynthate. So they're absolutely key in the growth strategies that plants use. Therefore, they've been used in many incidences to predict the strategies that plants can use to capitalize on these investments. So very commonly measured leaf traits include specific leaf area, as well as leaf nutrients such as phosphorus, potassium, and nitrogen, of course. So specific leaf area is a composite measure, a morphological measure, which takes into consideration the amount of dry weight tissue that is invested in the overall leaf area. So the leaf area is the surface that a plant is using to capture light. So from these relationships, from broad geographical relationships, 2 extremes of investment strategies have been described in the literature. On one hand, we have resource conservation specialists, which tend to have lower specific leaf area, lower leaf nutrient content, and lower photosynthetic rates. And on the other end of an extreme, we have resource acquisition specialists, so plants with higher specific leaf area, higher leaf nutrient content, and a higher overall photosynthetic rate. So if I can show that to you or illustrate it in a quantitative way, if we had a figure with specific leaf area on the x-axis and on the y-axis leaf nitrogen content, we would have leaf traits have been used as a comparative measure, so we'd have a resource acquisition specialist, so a faster growing species at the high end, and a resource conservation specialist at the lower end. Now these relationships have been determined by, were really kickstarted, they have a long history, but they were kickstarted in 2004 with a paper in Nature which described the worldwide leaf economic spectrum. And this was an incredible, groundbreaking initiative that brought together plant physiologists from around the world, ecophysiologists, and they brought together their data sets on leaves, and they found a remarkably strong relationship between leaf mass per area, which is the inverse of specific leaf area, leaf nutrients such as nitrogen, as well as assimilation rates, which is a surrogate measure for photosynthetic activity. Now, this study was a compilation of more than 2,500 species at more than 170 sites. It included leaves that had been measured in the Arctic tundra to the tropical rainforests, from hot to cold deserts, from boreal forests to grasslands. And it was definitely a strong relationship, but at a biogeographical scale. So long-term evolutionary processes and environmental conditions have sculpted the leaves in these systems. Somewhere along the way, and I have done this as well in my research, what has been interpreted is in these leaf relationships is that this also holds at a local scale in response to short-term perturbations. But yet, there hasn't been a large-scale experimental study on how that holds up in specific biomes such as grasslands. The information that's been tested has largely been observational studies or space for time substitution experiments, and there's been very few experimental tests of this response. And it makes it really difficult to draw these inferences and to start new communities based on leaf traits and based on these strategies without testing it in an experiment. So this brings me to the questions, and I'm going to show you some evidence that we found on these ideas in this experiment today. So just ask some simple questions. Do leaf traits and their correlations between these traits respond consistently to local scale short-term environmental change, such as eutrophication and a reduction in grazing pressure? And taking that a little bit further and building off of Angela's talk, how do the system level influences of climate, pre-treatment soil nutrient conditions, but also species temporal turnover affect these responses? So this study was conducted as part of the Nutrient Network study, which is quite a unique collaboration of more than 100 sites around the world where we put in the same experiment. But in the functional trait study, only 27 of the sites contributed leaf traits to the experiment. It always sounds like so much less. But the majority of these sites were in the US, but they also included sites from Europe, Australia, and also just a couple of sites in South Africa. And in the Nutrient Network experiment, individual principal investigators have established the same experiment. So at most sites, 3 blocks were established in a grassland. And in these blocks, there are 10 plots, and each plot has a different experimental treatment. And I show these experimental treatments in this area. And remarkably, it's the same over here. No, I'm just kidding. And each one, so there's a control treatment, and includes all factorial combinations of nitrogen, phosphorus, and potassium, as well as a fencing treatment. And that's where we fenced off these plots, which were 5 meters by 5 meters in size, to grazing by vertebrate consumers or herbivory that were more than 50 grams in size. And we also included in that herbivory experiment an addition of fertilizer, so either a control, no fertilizer, and an addition of fertilizer, but a complete fertilizer addition. So for the leaf trait measurement, what each principal investigator did was they collected 1 leaf from 5 individuals for the 3 to 5 of the most dominant species in each one of those plots. So it depended on the overall structure of the grassland, because these grasslands are wildly different. I'll talk about that in a minute. But also, so it depended on how many species were in that site. We wanted to capture the dominant species. And we chose different species depending on the plot, because we wanted to capture the response of those smaller communities in that 5 meter plot to the treatments themselves. So in some cases, the grasslands had seen a big turnover in response to different treatments even after 4 to 5 years, and in other cases, they had very similar species across those plots, but we wanted to capture that in the experimental design. So in total, we sampled more than 240 species and included grasses, forbs, and legumes. And we measured traits on more than 2,600 plants, and these traits were sent to Australia, and we then ran analyses for sometimes specific leaf area, but also leaf nitrogen, phosphorus, and potassium. So in total, our coverage of those sites represented species at 26% of the abundance in those plots. So here are some of our results. So these results I'm showing you here are the effect sizes from Bayesian hierarchical models that we solved with Laplace approximation. So it might be hard to see this from the back. It's a bit smaller than I thought. But on the x-axis, we have the treatments, so the 9 treatments, so nitrogen, potassium, phosphorus and potassium fencing, and the fertilizer plus fencing. And in this case, the 0 lines, which are shown in a bolded text, represent the control treatments. So any of the effect sizes, the mean parameter estimates, and the credible intervals that are above that line represent an indicator of a response, or below. So in the case with specific leaf area, so our only morphological leaf trait, we found very little consistent response across these sites to the nutrients or the fencing exclusion, which we would have expected based on existing theory. We did find a small response to where nitrogen and phosphorus were added, and also nitrogen, phosphorus, and potassium. So if nitrogen and phosphorus were together, we saw a very tiny response in specific leaf area, an increase. Where we did see a response was in to the soil nutrient addition treatments. We saw an increase in the respective leaf nutrient content. So where we added leaf nitrogen, we saw an increase in leaf nitrogen. Where we added soil nitrogen, where we added soil phosphorus, we saw an increase in leaf phosphorus. And where we added potassium, we saw an increase in leaf potassium. Now this result is actually quite surprising if you know the agricultural dilution effect hypothesis, which has been around for a very long time, that where you have an area open up to herbivory, if you add nutrients, you shouldn't, according to that long-standing theory, see an increase in individual leaf nutrients because of the growth diluting that response and the herbivory and the selective grazing. So that was quite a surprising result, even though it seems like a no-brainer on that one. So these are hierarchical models. So what's interesting about them is we actually separate them. We have random effect structure, and in the case of this particular study, how that variation is distributed in that model is actually quite interesting. So in this case, we had a random effect structure of block nested in site, nested in species, and because in some cases we had species across multiple treatments, but also across multiple sites. So to understand a lack of response by SLA to the treatments, some of it is revealed with these random effects in that you can see a fair bit of variation was explained at the site level, as opposed to the residuals, which are shown in red, and leftover variation to be explained by the experimental treatments. So there is more residual variation, but also with potassium, you can see an interesting amount of variation that's explained at the species level, whether or not a species will uptake potassium in response to increased availability. So as I mentioned or alluded to before, our grasslands are really different. So it's interesting to find a commonality, but it's also interesting to look at how that difference explains some of our variation. So we had grasslands with mean annual temperature of 0.5 in Switzerland. Switzerland always has to be the best. And then Australia as high as 18.3. So, very wildly different temperatures. Our mean annual precipitation was 250 millimeters up to over 1,800, both in the US. Of course, we had the lowest phosphorus soils in the study at 7 parts per million, and as high as 254 parts per million in Germany. So very different starting points. And soil nitrogen as low as 0.04% from the US and as high as 1.7% in the UK. So some of our leaf trait response could also be explained by compositional change in those plots, so species temporal turnover from year 0 to the time where we collected the traits. But also it could be explained by a plastic response of the resident species. So maybe those dominant species are just able to change their traits in response to that perturbation. So what we did to unravel that, and I'll explain this in a minute, so I don't expect you to be able to interpret it from this, but we used a structural equation model to try and piece together where that variation is coming from and the importance of the environmental characteristics as well. So what we found, for example, when we looked at leaf nitrogen, we found that our experimental treatment explained the response and it had a positive relationship. So the solid lines show a positive significant relationship, and the dashed lines show a negative significant relationship. And for example, we found that mean annual temperature had a negative relationship with leaf nutrients, which I think was a finding that Angela has found in some of her studies with leaf size, I think was it. No, it was someone else. So we see, and we also find that temporal species turnover has a positive relationship with leaf nitrogen as well, so we're getting a change in species in response to those treatments. And we see very similar results with leaf phosphorus, and very similar results with leaf potassium in many ways. There's some complexity there that I'm glossing over because of time. But then when we get to SLA, specific leaf area, it's doing its own thing. So once we take into consideration the other factors, there's no longer a significant relationship with our experimental treatments. And even mean annual temperature has a positive relationship, significant relationship. So what does this all mean? So I think with this first global experimental test, we show that the causal relationships do exist between fertilization and even in the short term. This is a short-term experiment with leaf nutrient content, but with specific leaf area, we did not see the response that we thought we would, and that potentially it requires a longer time frame, or potentially we need a changeover in species composition. We also found that abiotic conditions play a lot in our differences, even in one biome, the grassland biome. And I'm going to go out on a limb here, and I'm going to potentially suggest, I think my time is almost up, that perhaps some of the research that's been going on in leaf trait, and for leaf traits, but also potentially plant traits, could be compared to a phenomenon that was occurring in the 70s and occurred for 40 years in evolution called the adaptionist program, where there was a lot of criticism being put forth from scientists in different parts of the world that were evolutionary biologists who were explaining small changes, were finding adaptive characteristics to small changes in organisms. And perhaps we're at that stage with some of the traits that we've been measuring. So thank you.
No questions. If you think of any, you can ask Jen at the end. We now have Robert Kooyman presenting, who is an honorary research fellow at Macquarie University and an honorary research associate at the Royal Botanical Gardens and the Missouri Botanical Gardens.
All right, ready to go. Okay, thank you very much, and plenty to think about in the world of traits. So I'm going to talk a little more conceptually and a little more sort of hopefully leading to some practical insights into how we might use traits to understand more broadly community assembly, which is something that I do and focus on quite extensively. And as part of that, of course, we're looking at the way plants are distributed and their traits are distributed across gradients of environment. And those environments and those gradients, of course, can be quite steep, literally and figuratively, and of course the variations across those gradients can be quite evident. So just to remind ourselves, of course, that even at a smaller scale, we see quite remarkable variations across soil gradients. In the background, flooded gums and rainforest on basalts. In the foreground, Araucarian simple notophyll vine forests with Serrata petalum, and again in some of those instances, brushbox Lophostomen and New England blackbutt on exposed ridges. And if we put all of those communities together, of course, and we measure their traits, we see quite significant variation in trait values across those gradients. But within a particular biome, of course, we get the opposite effect, which is a convergence. So we get similar species converging and assembling into, say, a rainforest biome. So our expectation in general is that despite the massive variation in things like leaf size and within a single sample size, we still have a convergence of factors and traits, of course, in relation to species that assemble into single communities. So as they do that, of course, and Angela described beautifully the seed size conundrums, but seed size is still an informative and an important trait, of course. And it's not just the size. As you can see here, it relates to things like dispersal mode and the reward for the frugivore who in some cases is dispersing. And of course, things like the weight of species that are wind dispersed shapes the way they operate. So I'm going to talk very briefly about a convergence of traits here, so seed size more generally, but related to dispersal. Leaf traits, and I'll leave the leaf economics alone, and I'll just get you to think a little bit about leaf longevity and shade tolerance, because I'm going to talk about rainforests and those sort of species variations and differentiations find expression in the rainforest rather nicely because of the self-shading, of course, that goes on and the structure of the forest. Maximum height at maturity, that's already been signalled as a key component of dispersal capacity, but equally it affects shading. Wood density, that relates to how fast you grow and how long you live. So that gets back to linking with leaf traits and longevity and shade tolerance. And persistence as a trait, which is in situ persistence. And it's an extension of how long you live. If you live a very long time, you disperse a lot of seeds through time and you can occupy those places, but you can also resist invasion and competition by just holding the same ground. And some of the species that I study live thousands of years, and in some cases probably tens of thousands. So if we look at Australia, what we get then is the distribution of rainforest species across the whole of the continent. So this is not a map of the rainforests, this is a map of species distributions. And automatically, of course, what you get is something of a heat map about where the rainforests actually are. So what I wanted to demonstrate first of all, of course, is that the pool of available species, even within a single biome, is critically important and influences the assembly process, and also, of course, the range of traits and the variation in traits and plant strategies that are evident at a site. In Australia, it's not going to be news to you that the rainforests simplify as you go further south. So as latitude increases, we get a simplification, but it's not uniform, and it sort of stumbles around a little bit here and there. But and it also decreases with decreasing moisture availability. So those large-scale patterns, unlike those smaller patterns that we showed just a moment ago, also influence the available pool of species that can assemble in a particular site and be filtered into an assemblage. And there's another aspect to the Australian rainforest flora, which is its origins and its ancestry. And that, of course, includes the Sunda and Sahul, the Sunda being Indo-Malesian elements and the Sahul component, which are the Gondwanan component. And of course, we've just done and published some recent work on how that is filtered, and you can see there very briefly the extent of the Sunda invasion into Australia, and during the climatic variations over millions of years, the expansion, contraction dynamics of rainforests on the continent, it's made room for species from Sunda to merge into Sahul, and also the other direction. So in that case, the results of that research work showed that smaller seeded, fleshy fruited taxa were more likely to be dispersed into Australia from Sunda as a consequence of the dispersal mode, which is predominantly birds. So when we come to that, of course, and seed size is a critical element, but it doesn't tell us everything about how plants move around. And some plants move very successfully, and we've got an example here of Elaeocarpus grandis or angustifolius, which is genetically, as we now know, is a relatively recent arrival in Australia and has very successfully expanded its range, courtesy of wampu fruit doves and a few other dispersers. And then we've got, and its origin, if you like, is Indo-Malaysia. And then we've got another species in the same genus, same family, of course, Elaeocarpus sedentarius, which has a similar sized fruit. It looks superficially the same, but when we come to understand what's going on, in one case we've got a fruit with a wonderful reward for the frugivore, and on the other case we've got one with no reward for frugivores, but a nice rich oily seed which attracts predation by rats. So in the end, one is moved around locally by rats and the other one's flown vast distances by our premier wampu fruit dove disperser. So very different outcomes for superficially what looks like similar taxa. That's not universally the case with Sunda and Sahul species, but it just allowed me to bring that sort of story of origins together with trait variations and strategies. So when we bring Sunda and Sahul together, of course, which we do in the far north, in northern New South Wales and southeast Queensland along the Lamington, where we get side by side the so-called subtropical and the so-called cool temperate. Don't ever use climatic typology. This is a simple microphyll fern forest sitting next to a complex notophyll vine forest. One has lianas and many taxa with Indo-Malaysian origins, and the other one is Gondwanan, sensu stricto, and very much simpler. So reflected in the rainforest, of course, are plant strategies, and those strategies really are about looking at fast versus slow lifestyles, and to understand how that operates and how we see that working in a height, light and competition-based rainforest assembly process. I've removed one of Dan Foster's fantastic little model drawings here, and just to demonstrate how early succession species have pretty much all the same strategy. They're all competing together. And they have relatively low tolerance for competition. But as we move into other species that grow taller, that live longer, that are more shade tolerant, we get an increase in diversity and the spatial distribution of those species, and we get a movement towards a more mature community assembly. So height, light and competition influence rainforest assembly, and that varies, of course, in relation to the size of the patch, the size of the gap in the rainforest, and is influenced, of course, and links to seed dispersal and seed rain. And that's, I guess, what we're looking for in terms of a restoration outcome is to some extent to replicate the structure and diversity of the original forest. And so planting designs then should, in general, take account of landscape context, the need to plant at all is the first question, and then followed by the likelihood of attracting in dispersers and diversity through time. And if those likelihoods are low, then you're in the business end of planting species in mixtures that create the structure and diversity, the phylogenetic spread, if you like, the taxonomic spread that's of the original forest, but also create a structure that enhances all of those different components that you can see there. The low light levels at ground level, the accumulation of litter, the canopy closure, the shading, the differential height gain by virtue of shade tolerance, the range of life forms, but also, of course, leaf sizes, seed sizes. All of those sorts of things converge by using that, allowing traits to inform your decision making in terms of plantings and management of locations. So just to remind ourselves, that's again the tropical forest canopy. And basically we'll just run through what I hope was clear, that basically traits do reflect species lifestyles and their capacity to operate in the rainforest habitat. Biogeographic history filters and delimits the species pool, and that species pool is filtered into assemblages by environmental gradients. It provides part of the explanation of high diversity in tropical forests and subtropical forests, and competition and competitive effects generally have been suggested as operating more strongly among more similar species, that is, more related species. However, species can be more tolerant of competition as well, and so it's not just a one-way thing with competition, that species that are good competitors can actually tolerate a little more competition and persist. So that could also be a major factor in promoting coexistence and result in both higher abundance and higher diversity. As I said earlier, there's a convergence of factors that and trait similarities in similar habitats, but there is still a trait-mediated niche petitioning and differences in dispersal, establishment and growth during vegetation development filter the species. So we've got Sunda and Sahul components deep in paleo-ecological time, and the movement of Sunda and Sahul taxa in both directions to create the species pool. And that's facilitated largely by both seed size and dispersal capacity that reflects the availability and reward of fleshy fruits. And in the end, we find that traits can shape community assembly and species distributions, and they can inform restoration efforts, but not always.
Thanks, Robert. We now have Jarrah Wills, who is a postdoctorate research fellow at the Queensland Herbarium.
Good afternoon, everybody. My name is Jarrah Wills, and today I'll be presenting two case studies from my PhD, which I did with Jennifer Firn. And they'll both, I guess, give some insights into ecological processes that might or may not be useful for tropical reforestation. So I'll just be focusing on tropical forests today. So the first case study that I'll be presenting is from the Philippines, and the second is from North Queensland. So firstly, just to give you some background, so both these two regions actually have quite a lot of similarities. There's the respective English and then Spanish and American colonization events has led to huge amounts of land clearing and a shift to sedentary agriculture. And that shift was then accompanied by declines in productivity, and then that land was eventually abandoned. And so there's huge amounts of degraded land in both the Philippines and North Queensland. So there's a really big need for improved reforestation. The ecological processes we were looking at in the Philippines was that of seedling recruitment, and in North Queensland it was tree growth. So I guess the key question from this symposium, and hopefully my talk will address some of it, are can functional traits help, and can they, I guess, highlight ecological processes that can then be applied for practical reforestation. So firstly, to the Philippines, what did we do? Well, the question was, what was the differences in recruitment between different forest types? So the forest types that we used ranged from simple monocultures to mixed native species and then to secondary forest. And there were adequate replicates of all of those forest types. So we sampled all plants below 2 metres in height, and so that was identify them and work out their relative abundances. And then from that we created a plant functional database where we got reproductive traits, which we chose to use, and extracted them from databases, books and literature sources. And that include dispersal type, fruit size and seed size. So what did we find? Well, in the Philippines, we found, just using pure species diversity, we found a much significantly lower number of lower diversity, within the monoculture plantations, and this is what we expected to find. And so the mixed species plantations were intermediate between the two, and then the secondary forest was the highest. However, we really started to get some insights when we used functional trait diversity. So using dispersal type, there was significantly lower number of wind-dispersed species within the monoculture forest types. And you can see that in the bottom graph here. On the y-axis is the number of species that are wind-dispersed, and forest type is on the x-axis. So you can see monoculture was significantly lower for this functional group. So this is dominated by the family Diptocarpaceae, but is not limited to that family. It also includes numerous others, which are often tall, well, just about always tall, straight emergents, and they're often targeted for harvesting, for logging operations due to that beneficial timber properties. So again, using functional traits, we also found using fruit type, the similar number of species with a syncarp fruit type. So this was dominated by the family Moraceae and includes the figs, but also several other Artocarpus species. So these species were able to colonize out into the monocultures and recruit into those forest types. And you can see that on the top graph there. Monocultures were quite very similar to secondary forests for this fruit type. Another insight we got using functional traits was in the largest fruit size class, there was significantly higher number of species within that class for the monoculture forest types, and this was counterintuitive from what we predicted. And this was, when we looked at what was making up that relationship, it was dominated by domesticated species, so species that had been dispersed by humans into those plantations, and that included things like breadfruit, jackfruit, pomelos, those sorts of species. So we got some quite in-depth insights using functional traits for this data. Now to the North Queensland example. So totally different area, totally different study and different question. And the question we asked was, can simple leaf traits predict growth rates in diverse tropical forests? So can you go up to a tree or a sapling, measure some really simple traits of its leaves, and will that tell you something about how fast that tree's been growing? So it would be pretty useful for practical reforestation. So can you do it? Well, to answer that question, we used a really rich historical data set which dated back to the 1940s and 50s, and we used that data to get estimates of tree growth. And after that, we selected for the trees that we wanted to go out and collect leaf traits for, and you can see on the right-hand side that's a stem diagram of the plots we used in North Queensland, and the black dots are trees where we went out and collected leaf traits. How did we collect leaf traits? You know, basically we used a really big slingshot to pull down leaves from the canopy, and we selected, did multiple branches per individual, and this was in order to get adequate replication for each of the individual trees. So we then aligned that leaf trait data with the historical data and look for relationships. So what did we find? Well, in general, leaf traits were poor predictors of growth rates for canopy trees. However, as you drop down into the subcanopy, those relationships became much stronger. Well, and for the top graph there is periodic annual increments, so that's a measure of growth on the y-axis, and then leaf phosphorus, so a leaf trait of leaf phosphorus. And you can see for the subcanopy, that was a significant predictor of periodic annual increment for trees occurring within the subcanopy. That did not hold up for the canopy trees. So for subcanopy, we also found that there was stronger trade-offs between traits, so correlations between leaf traits, and these adhered much more to our theoretical predictions for trees occurring within the shaded subcanopy. You can see in the graphs B, C and D there, for subcanopy trees there was much stronger associations between these different leaf traits for subcanopy trees. So from this data we concluded that there's a really strong size dependence of traits and growth, and these really need to be considered before they can provide practical applications to reforestation. So here are some practical implications of the Philippines work. So firstly, there was a lack of wind-dispersed tree species across the fragmented landscape, and we were able to pick that up using functional diversity. We were also able to pick up that figs were recruiting across the landscape, and this also, it was interesting to note the domestic species that were being dispersed by humans, and we were able to recommend species to target, so there were direct practical applications that we could recommend for people undertaking reforestation in this region. So there was strong practical implications. So, and it was also analogous to other tropical forests in different parts of the world. So in Queensland, you can think of the tall wind-dispersed trees such as the booyongs, flindersias and red cedars. They're often really harvested for their beneficial timber properties, and that has been corresponded into, I guess, a lack of recruitment out into the open areas. So for North Queensland, what are the practical implications here? Well, firstly, it was difficult to provide practical recommendations for reforestation. We did get some valuable insights theoretically. However, to recommend species or traits that could be directly applied to plantings, we found it quite difficult. We also, I guess one application that we more of a caution is the use of assigning mean leaf trait values and using them to design reforestation projects. Because of that size dependence of traits, you can get leaf traits from multiple canopy stratas which might throw out that species mean. And also, in conclusion, I guess there's a lot of things, plant axis of variation that determine growth, and also leaf traits, and this is complex. So to wrap up, well, what is the utility of traits for tropical reforestation, and should they be considered the holy grail? From the Philippines example, it provided additional insights into plant recruitment, and it provided direct practical recommendations, and we were able to list species that can then be taken up and utilised by practitioners. In North Queensland, there were some theoretical insights, but they lacked direct practical recommendations. So I guess back to the question, can plant traits be considered the holy grail? Well, maybe not. They're definitely insightful, but they give sort of a more in-depth perspective into plant function or into ecosystem function, which can be applied, but it's definitely not considered the holy grail. In conclusion, I guess the questions, aims are important, and also what traits you use are quite important, and these should be considered when thinking about the practical implications for reforestation. Yeah. Thanks very much for listening.
Great, thanks, Jarrah. Well, we have time for some questions. Yes?
Do you have any information on wood density for those species? I wonder if I were to form the growth rates in your old tropical trees better than the leaf trees.
Yeah, so we did use dove-based values for wood density. The problem is, in forests, wood density varies quite a lot within species as well. And also, that did be a good indicator of survival, but we didn't find any correlation between growth rates and wood density for that subspecies. So, any more questions? Yeah.
Is there any way that predators or parasites might affect how traits are expressed? Because I recently went to Africa for a trial eucalyptus there, and the eucalypts were completely differently, and I got told if someone's with a predator, that changes the way eucalypts grow here. Because it affects the way traits are used, could that interaction affect this kind of as well?
Yeah, so I guess eucalypts in Africa, because they don't have the species-specific pests and pathogens like they do here, they're able to grow a lot better and faster, hence why they use them in forestry. I guess that is expressed in their leaf traits. I haven't looked at that in this study, but yeah, that's definitely... Yeah.
You said you were able to make recommendations in the Philippines, but what were those recommendations? Not specific, species, but what was the rest of the recommendations and how the traits help that?
Yeah, so it was, I guess the major practical recommendation was to, recently it was, so from the literature there's been a lot of talk about large fruited species lacking in recruitment across the landscape. So from our data, it suggested that also those wind-dispersed species should be emphasised in restoration plantings because they are over-harvested due to that emergent layer being exploited for timber production. So I think in Asia, the dipterocarps dominate about 70% of timber production. Yeah. Yeah. Sorry? Yeah, so for canopy trees, that association between leaf traits and growth wasn't very strong. And that was expected. It's been brought up in other studies. There's a strong size dependence of that relationship. And what we found is as you drop down into that canopy layer, into the shaded canopy, that association becomes much stronger.
Okay.
Thanks very much.
Thank you, Jarrah. Our next speaker is Carla Catterall, who is an emeritus professor at the School of Environment in Griffith University in Queensland.
Okay, well, I'm going to touch on some of the themes already addressed. In a slightly different way, because I'm going to focus my talk within a specific kind of ecosystem within Australia, and that's what I'm calling the heritage moist rainforests. And these are I suppose, you might say, the jewel in the crown. Very structurally complex, full and layered. And I'm going to look at the tropical uplands and subtropical lowlands, which actually have very similar climates. That's what Rob was saying, these terms, tropical and subtropical, are not really useful in this context. They also have a lot of shared plant and animal species, and it just reflects a wider prehistorical extent of rainforest. The rainforest in Australia has shrunk, and biggest bits remaining are in the subtropical lowlands and the tropical uplands of this type. Additionally, when European colonists hit the scene, they set about clearing the forest. The top two pictures are from North Queensland, the bottom two are from the subtropical rainforest landscape in the Big Scrub, which was particularly heavily denuded. Those bottom photographs show the situation in the 1950s. It's now got somewhat better, actually. But anyway, there was not much forest left, and it had been converted to a totally different ecosystem type. That is, introduced pasture grasses, mainly seafall grasses from Africa, with a lot of grazing livestock, particularly cattle. So the current challenge, which is ongoing, is to replace more of that pasture with restored rainforest biodiversity. And the fact that we're dealing with this diverse rainforest ecosystem presents opportunities and challenges. The high diversity of plants and animals is an opportunity that, there's a lot of Australia's diversity in these systems, but it's also a challenge to be able to restore that number, that diversity in the system. And we're also dealing with taxa of high conservation value in that they're representing ancient Gondwanan lineages and endemic species. The other characteristic, which is both an opportunity and a challenge, is that these ecosystems feature a lot of interactions between plants and animals. If you consider the life cycle of a plant, or in this case I'm talking about trees that produce seeds,
They have to be dispersed, the seeds will germinate, become seedlings, they have to survive in order to do that. Seedlings have to grow, and in order to do that, they have to survive. And interactions occur firstly in the dispersal stage with frugivores, and secondly in the seed and seedling stage with seed predators and herbivores, as well as the role played by the physical environment. And so the issue is, how do the trays of plants intersect with those processes in order to enable forest to regenerate? And I guess the attractiveness of looking at trays is that when you're looking at very large numbers of species, if you're dealing with a couple of hundred species, nobody's going to study each of those in detail. But if you can find some trays which actually indicate performance in different kinds of species, then all you have to do is figure out how to classify each species ecologically in order to know how it's going to behave during restoration. And I'm particularly going to focus on dispersal processes. They're really best understood. You've already heard a lot about that. And in these rainforest ecosystems, the majority of, it should say rainforest trees really, are fleshy fruited, and they use animals in order to disperse their seeds. And what that does, what that co-evolutionary interaction has done, is give us a beautiful array of fruits. So beautiful, in fact, that we have renowned artists producing books that illustrate the fruits of the forest. Here we have the Wet Tropics Manual for Rainforest Restoration, and you can see that fruits feature here. What are people but large fruit dispersers? Primates are important fruit dispersers, and we're increasingly seeing the value there. So restoring rainforest involves restoring that diversity, that functional diversity of fruit types. In terms of seed dispersal and these kinds of forests, worldwide, the fauna, the dispersers, really, I think, have been better studied than the trees in an ecological sense. And it's in the last decade or so, it's really become clear that around the world, what is happening is we are losing the larger bodied seed dispersers due to a variety of processes that include deforestation, fragmentation, and in particular hunting. So if throughout Asia we have what's now called the empty forest syndrome, where you don't have the large-bodied seed dispersers anymore. Well, in Australia that's not so much of an issue. We don't heavily hunt our fauna. But even so, we do have the effects of clearing and fragmentation, and this is based on the work of my PhD student, Kath Moran, several years ago. And this is in the subtropics, and each point on this graph represents a bird species that is a disperser of seeds. And what this vertical axis is representing really is the ability of each species to disperse larger seeds. And in this case, larger seeds really it's the propagules I'm talking about, and the propagules being more than 10 millimetres in diameter. And this group of species here are those which are more or less confined to intact, reasonably large areas of intact forest, better developed forest. These ones on the right are the ones which rove around in the cleared country pretty well. And so the ones that live only in the forest basically are better able to disperse the large seeds than the ones that are out in the open country, where you really want them to be moving seed around. They're less likely to be eating the larger propagules. And so we have most dispersers outside of intact forest consume mostly small fruits. Does this mean that larger seeds do not get dispersed? So to look at this question, first of all, I'm going to look at the idea of this propagule size and seed size. So the thing that's getting moved around by the frugivores, which are mostly birds, are being called diaspores, and the diaspore size is the diameter of the unit that gets moved around. And this is the size frequency diagram showing the number of species from some quantitative sampling in the tropics and subtropics of different diaspore sizes. And even within, say, a genus, you get quite a lot of variation. So here we have one syzygium, here we have another syzygium, and here we have a third syzygium. And for these, the seed is encased in flesh to attract the seed disperser. And when I talk about the diaspore size, I'm measuring the diameter of the whole thing. And you can see that between about 6 and 10, 11 millimetres is a common size. Down here, these hatch bars represent fleshy fruited things, because you can't get some of this stuff just by looking at the seed size as defined by taxonomists or the fruit size as defined by taxonomists, because they often use funny definitions that don't correspond with the ecological function. So you have to reinterpret all this from the sources. So here, this diaspore size is the aril surrounding the seed there. Anyway, I won't go into too much detail about that, but these are diaspore sizes that exceed the gape width of most birds that we now have that can fly. And so what disperses them? Well, cassowaries or maybe emus, or short distances by mammals, and maybe once upon a time by the now extinct megafauna. This is the same set of species, and now we're looking at actually their seed size. Over here on the left we have figs and picturus that are very soft fruit that can be pecked into very easily, and so in this case the diaspore and the seed are the same thing. So we have a variation from very small, 0.01 of a millimetre across, to the maximum of about 75, I think it is. And these are all seeds that have a frugivore attractant. If we look at wind-dispersed species, which are recognised according to whether they have particular structures that appear to assist them in wind transport, you can see that their seeds, and in this case I'm only looking at seed size, and I'm only looking at the seed, not the entire fruit, the seed diameters are smaller. And again, we've got here, this is a large species of Agarhodendron from the wet tropics, and this is a smaller species from the subtropics, quite a range within particular genera. And there's 31 species that have other dispersal modes. The black bean there on the right, which I won't say anything more about. We don't really understand how they're dispersed. And they're a minority of species. And this big line here represents the divide in frugivores. So to the left of that, there are frugivores all across the landscape that can disperse those seeds. To the right, it's only those, or primarily, much more dominated by bird species that are confined to the rainforest. Okay, how much more time have I got? 3. So what about when we regenerate in these landscapes of disused pasture? We want to get from pasture to forest. I think I'll skip through this. We want to become more similar to the forest, and our indicator, or my indicator, is the diaspore and seed trays of these species. And we could use different kinds of interventions, and I'm going to look at 2. One is really not much of an intervention except for reducing livestock grazing, which allows regeneration to take place on its own, and the other is more intensive tree planting. And to look at this, I'm taking more established sites. They've got a canopy, they've got trees there, aged 11 to 15 years for, I've got this the wrong way around, sorry, aged 12 to 59 years for the regrowth and 11 to 15 for the plantings. what happens with seed size diversity? Well, this is in our reference rainforest sites, and this is in North Queensland, and I am choosing to look only at native tree species that achieve a height of maturity of at least 10 metres. So I'm looking at bigger, not the little stuff, but potentially bigger trees. And also, if we take our survey data and look only at what I'm calling the mature stems that are around about 10 centimetres or more diameter, this is the percent of species, large seeds. So roughly 44% of species of stems have large seeds, are species that have large seeds. And this blue says that they're bird dispersed and the purple says that they're wind dispersed. So that's what it's like. And this is the rest of the species, the smaller seeds, and they're mostly wind dispersed with mostly bird dispersed with some wind and some of the unknown. And all of these add to 100%, okay? So that's our forest. What happens in regrowth? Well, almost all the stems in the regrowth, reasonable diversity of species, but they are small seeds dispersed by birds. But that's for the established trees, okay? Now, if it's moving in the direction that we want it to move, you might think, well, what about the smaller things that have come up more recently? Maybe they represent more large seeds. So to look at that, we take what I'm calling recruits, things less than 2.5 centimetres diameter. And in the forest, it's not too different. The regrowth is just as dominated by species with small bird dispersed seeds. So it doesn't look like it's tending to get better over time if you look at what's coming up. Now, when we plant trees, we overcome that by planting a diversity of species. In fact, people plant quite a lot of small seeded wind dispersed species relative to the forest. So the plantings have a relative over-representation of small seeds, but they're certainly a lot better than the regrowth. What about what's coming up in the plantings? Well, we seem to see a shift towards more small-seeded bird dispersed species. So, from that I'm concluding that seed size is predicting and driving recovery processes. Tree species with larger seeds disperse less and regenerate more slowly. That's where I stop trying to explain things, okay? I don't claim to understand that. Dispersal is part of it, but seed size is also related to many other traits that will drive the survival and germination of the seeds and the growth of their seedlings. And I don't understand how all those things interact with each other. But if you're a restoration practitioner, I would turn it into some questions for you. When you do the weed control, is it leading to increased regeneration and diversity of the larger seeded trees? When you plant framework species in a planting and assume that the frugivores are going to bring seeds in, is that what's happening? In general, if what I'm saying really does have wider applicability, what are the other creative interventions? Are there any, or do we just have to wait? Why is this happening? And how does all this relate to species successional categories, which are often used, but which I've avoided mentioning? Thank you.
Great, thank you, Carla. Our final speaker is Daniel Laughlin, who is an associate professor in the Department of Botany from the University of Wyoming.
Whoa. Somewhere here. There we go. All right, ready to go. All right, well, thanks for coming, everybody. This morning I talked about a model that we can use in restoration to translate traits
into species assemblages to test some fundamental ideas in ecology, and that's what I'm going to talk to you about today. So the problem we face with restoration is that if we only focus on historical assemblages, we run into some problems when we're trying to set our restoration targets. Often those historical conditions are unknown and cannot be really clearly defined. And right now we're living in a time of very rapid climate change. So if we base our restoration targets on conditions that were created hundreds of years ago, then we're going to set ourselves up for a possible failure if we're trying to restore communities that won't be able to tolerate the conditions of the future. Okay, so what can guide our restoration efforts that is anchored in ecological theory? And so what I argued was that traits or properties of these species provide us some information about their ability to tolerate environmental conditions. And if we can match traits to these environmental conditions, then we might be able to improve our restoration success. So whether we base our restoration targets on historical conditions or base them on future conditions, we can view both of these through the lens of the traits of those species because they evolved to adapt to those conditions. Okay, and I talked about these new models this morning, and that's what I'm going to talk about today. We're going to dig into what these new models actually do. Okay, so one thing I want to say right up front is that managers don't manage traits. Okay, this is an important thing. These are just attributes of a species, but managers are masters at manipulating species. So as an ecologist, our job is to be able to create a tool that can translate these traits into species assemblages that can therefore be manipulated. Okay, so at the very simplest, if you remember one thing, what this model does is you input traits into the model, and the output is a species relative abundance distribution that satisfies those traits that you specify. Okay, so what we're going to be doing is focusing on the arrow right now. What is this model actually doing? Okay, so this is a quantitative solution based on a system of linear equations. I'm going to just walk us through this step by step. So the first equation is that the sum of the abundances of the species, they all sum to 1. Right? That's the P sub i's. They all sum to 1. Okay, so let's think about this for 3 species. Okay, often we're dealing with dozens of species, but for simplicity and to illustrate it geometrically, we'll just think about 3 species. So this is 3 axes that represent the abundances of 3 different species. So this first equation, when we specify that first equation, that constrains all of the possible solutions from this infinite 3-dimensional space down to a single plane, this gray triangular plane. So all of the spots that are located on that plane, they all represent solutions where the abundances sum to 1. Okay? Now the other equation we want to add is the second equation, which states that the linear combination of the traits and those abundances, they all sum up to this average trait value, this community-weighted mean trait value. Okay? So then if we add that second constraint, that further constrains all the solutions to be located on a single line that occurs on that plane. Okay? So we have the 3 species. That's the trait value of species 1 attached to its abundance. That's the trait value of species 2. You might think of them as leaf nitrogen concentrations. These are on the scale of leaf nitrogen. And that's the trait value for species 3. And what we've stated is we want a community that has an average trait value equal to 2% leaf nitrogen. Okay, so then we can solve this solution, and we see that all the values along that line satisfy these trait values. Now, we can think about that. That's a lot of different solutions, and you can think about that as a range of variability, which we like to think about in restoration, some variability in nature that we want to mimic. But oftentimes we just want a single solution or an average solution to talk about. And Bill Shipley suggested that if we wanted to do that, one way we can do is to find the solution that optimizes the entropy of the system. Or you can also think about it as Shannon's diversity index, which is equivalent to the entropy function. Okay, so I talked about this morning about an example where if we wanted to restore resilient western U.S. conifer forests, we can think about the challenges that we're facing. One of the challenges is increasing drought and increasing fire frequency. And if we can translate those challenges into looking at the traits that might lead to success in these future conditions, we can look to two traits in particular. One is wood density. Now, wood density has been correlated with a physiological trait called P50. P50 is the xylem tension at which 50% of your vessels become embolized. So it's indicative of your tolerance to drought. And for both angiosperms and conifers, wood density has been reasonably correlated with this tolerance to drought. Also, thick bark will promote resistance to surface fires. So as your bark thickness increases, your probability of survival, or probability of mortality as shown here, really decreases. And in the systems that we're working with in western U.S., you can get trees that have bark that's like 10 centimeters thick. Okay, so super low mortality in surface fire. So if we want to create a community that has both dense wood and thick bark, then we can enter those as those constants in those equations on the right-hand side of those equations. And if we do that, we enter our species pool, the traits of the species pool into the model, and we come out with the abundances of those species that satisfy these conditions. And in this case, for the desired future conditions, it said that ponderosa pine, Douglas fir, oak, and maple should have pretty high abundances if we want those species to survive in a thick bark and or a dry and frequent fire system. Another thing that's useful is to test the idea that, well, what if we increase the size of the species pool, bring in species with traits that might even be more drought tolerant or more fire resistant from other regions of the country? And what we find is that we see combinations of species that don't exist in contemporary ecosystems. Okay, so we actually can create, using this model, we can create no analog systems. So combinations of species that currently don't exist, but that might exist in the future and live in these sorts of conditions. But there's another problem. Okay, this model basically selects species that are functionally similar. And one of the long-standing challenges in restoration is to restore diverse communities, right? Not only species diverse communities, but functionally diverse communities. So how do we do that? Well, one possibility is to optimize a different function rather than entropy. You can optimize Rouse-Q, or this is a popular functional diversity index in ecology that is a nice closed-form algebraic solution which you can optimize using some linear programming. But if you do that, the solution falls at the bottom of the floor of this figure. What it's stating is that if you choose that solution, you're going to increase the abundance of species 1 and species 3 and reduce the abundance of species 2. Because what this functional diversity index does, it selects species in the extremes of trait space and then says, okay, we don't want anybody in the middle of trait space. So that's to the detriment of species 2. This is problematic. So one of the things we did to try to fix that is we tried to get what we really want is an even distribution of those traits to really optimize functional diversity. Okay, so to illustrate this a little more, what I've done here is I've created 16 species that are located in evenly across trait space for two arbitrary traits, so trait X and trait Y. And what we can do with this model is, say for trait X, we want to select species that all have high values or have a high average value of trait X. But we want to diversify the value of trait Y. So if we use just a Rouse-Q, or that functional diversity index, what we see is the species in the middle of the trait y-axis all drop to 0. Okay, so we're selecting species at the edges of the trait y-axis, but the trait x-axis we're selecting species that have high abundances of trait X. But we want this to be more even across the trait Y value, and what we can do is just really, we change the diversity or the objective function to include entropy, include the evenness of that distribution. We can improve it even further by capping the distance matrix. And so what we have is this really nice, even distribution of species across the Y, and then species with large trait X values are then selected. Well, what would this look like for a real system of species? Well, here, working with John Dwyer, he has a nice set of specific leaf area and wood density measures from subtropical rainforest species. And these are all the species in this data set. And what we thought was, well, let's see what it looks like if we want to optimize high values of specific leaf area so that all the species that we're planting have relatively fast growth rates. But then we also want to diversify the values of wood density, because some species will grow slower with more dense wood, and that will start to stratify